So this is a different disease than autism, but there's a lot of similarities. And what also might be useful are some of the approaches that we've been trying to work out for this disease that would be looked at in autism. And so the basic approach is that when we started with this disease, we really didn't know much about it. And I wrote several grants to NIH which were turned down based on the fact that I didn't really have a good hypothesis, and then I explained to him that you don't understand the scientific method. The scientific method is observation, hypothesis, and a test. And if you don't have observations, you can't generate a hypothesis that's worth testing. So step one, generate a lot of data. Then look at that data and come up with hypotheses. Secondly, post all of that data on the web before publication to let other people use that data to generate their own hypotheses. Unfortunately, that wasn't sufficient to get a grant. And so I set up a charity by Linda Tannenbaum. It's called the Open Medicine Foundation. The Open Medicine referring to the fact that we're going to try to make everything available to everybody of the publication. So this has a complicated name. I usually just say MECFS. And the reason we have two names is ME is the name used in Europe, and CFS is the name used in the United States. And there's been a huge debate amongst the patients which you should use, and I just say, I don't think it's worth having the debate. Let's just call it MECFS and get on with it. We need to figure this out. This is a picture of my son. This is to dispel the notion that this is jumpy flu and it's not very severe and you're just tired, and meeting people who say, oh yeah, I had that once. I was tired for a few days. That's not MECFS. This is extreme fatigue. He is now worse than it is in this picture. He cannot eat. He cannot swallow. He has to be tube fed with a tube into the top of his intestine, and he has to be fed with an IV that a tube goes into his heart, and that's where he gets his fluid. He's super sensitive to smell and sounds, and he cannot be touched. It's very hard to take care of. I was a little late getting here today because he rang the alarm and he needed another saline drip, so I had to hook all that up. You see some bottles of water. That's when he was drinking water, but he can't drink anymore. So, but he doesn't want us to take it away, and that has to do with some of the anxiety. He doesn't like change because if it's changed, he has to think about it, and that causes us to use up too much energy. We have to go into the room all wearing black clothes because then he doesn't have to interpret the color. So the basic plan is that let's see if we can't find a biomarker. That is, this disease, like autism, is diagnosed with symptoms, and there's a danger of that you wind up with a lot of people that meet the symptom criteria but actually don't have the disease. And then if you go into molecular stuff, then you kind of pollute your database, making it much more difficult to understand what's going on. You would like to have a pure collection of patients. We would like to not focus on just getting a publication. That's what NIH wants us to do. They judge success as the number of publications. We want to focus on doing something for the patients. And I think that's true for this group as well. Publications are useful, they have to be published, but those publications ought to be trying to focus on what you can do. So we have three things: find a treatment that will make them better, maybe small, but if it helps, let's try to figure it out. Can we cure this disease? And we want to not say it can't be cured, we want to cure it. And then if we can cure it, what we'd actually like to do is prevent it, then we don't have to cure it. But I want to help the patients that are currently with it, so that this is sort of the list of how we focus on. Now, a biomarker in this disease is extremely important because a lot of physicians say there's nothing wrong with them. Now, these are patients that are able to go to a clinic and get evaluated. Many of them can't go. They're bedridden, and therefore they go undiagnosed. There was a study at NIH, sorted by NIH for the Institute of Medicine, which estimated that probably 80% of the patients are undiagnosed. This is a fairly common disease like autism. It's about 1% of the population. I get upset when I look at resources being spent. They ought to be a little bit proportional to the number of people and also the severity of the disease. The NIH should try to help the population, not focus just on getting publications. It's easier to get publications when you know a lot about a disease. So it kind of follows fashion as opposed to responsibility. So I showed you a picture of my son. If I go in and take blood samples from him and take it to a clinic and have those run, these are the standard tests that are run on everybody when they go in for their physical. The doctor can look at those test results and say, wow, this individual is in the peak of health. There is absolutely nothing wrong with him. Low heart rate, low cholesterol, everything looks fantastic, except he's bedridden. So therefore, we needed to have something to say, these people are not healthy, there's something seriously wrong. So that's the first pass, distinguish them from healthy, not necessarily from all of the diseases, that's a more complicated problem. So we tried this technology, which was something that we developed for another purpose. And it was in our efforts to develop diagnostic tools, and those tools try to make them cheaper, smaller amounts of material and so forth. So this is nanofabricated. We do a lot of nanofabrication at Stanford. We're not at state of the art. We're down to about 16 nanometers in our fabrication, and Intel's around 6. So, but we can still make some tiny devices. This device is small enough that you have a hard time seeing it. And we make them on a wafer so that we can move it around, and we have multiple devices on one wafer. But it's made with silicon, and then we put layers of gold, and these are little gold electrodes, and they're small. So it looks like a needle, and we call it a nanoneedle, and it has 2 electrodes in it, and they're insulated. So this is very much like your electrical wire from your house. There are 2 electrodes that go plugged into your wall. And what we're measuring from this is we can put a current through this and measure the electrical properties of anything that it touches. And we make, this device is not just one of these devices, because you can get false readings when you have one. We have thousands of them. It's a small channel, and thousands of them go into the channel. And we take a measurement about 200 times a second. So this is unlike what a lot of clinical tests where you make one measurement and try to get an answer. We make hundreds of thousands of measurements and take so we're much less likely to get a false positive or false negative. If you look at the results from this, the blue are healthy people, volunteers that have come to Stanford and donated blood, and the red lines are the patients. Now, here's what we reasoned to make this test work. This was just totally guessing, absolutely guessing. And that is, we said the patients are fatigued. Maybe their cells are fatigued. Well, how do we test it if they're fatigued? And one of the physiologists said, well, add some salt. Sodium chloride. If you add salt, they have to pump it out. So these are the immune cells. And you give them some salt, they pump it out. That pump has to have energy. And in order to pump them out, they have to expend energy. So you're effectively exercising. And you simply raise the salt concentration and see if you can see a difference. Healthy cells pump it out fine for hours. In CFS cells, something really happens to them. And how we detect something happen to them is measuring something electrical property called electrical impedance. Now, you don't need to know what that means. It is a complex measure of lots of different electrical properties that is extremely sensitive to some change in the cell. The cell can change size, it can change lipid disposition, it can change things inside of it, it can change structure. All sorts of things will change the impedance. So we don't know what actually happened. We know something bad happened. Now, this is 20 patients. We've done another 20 controls and 20 patients. It looks the same. That's a validation study to make sure we didn't do something wrong here with a new batch of chips. Probability that this is random is 1 chance in a billion. That's a pretty good diagnostic test. That's a lot better than a lot of other tests. So now we use that. Every patient that comes in, we run that test. Now we also like to look at some properties of what's going on, and this is blood. Now, the blood we take is blood minus the red cells. I won't get into why we remove the red cells, but it gives us a funny signal, so we get rid of them. And what we ask is this a property of the cell, or is it a property of the plasma that the cell is in? And so we do a switch. We take healthy plasma and put it on ME-CFS cells, and we take healthy cells and put on plasma from the patients. It tracks with the plasma. So you can take a healthy cell and expose it to the plasma from a patient, and they look like it looks like the patient. We do not know what it is. Now this is classic biochemistry. You fractionate and try to figure out what it is. The only problem is that it takes money. And so that's put on the shelf for the moment, but we know exactly what we have to do. We've also looked at the red cells. These are the cells that we were throwing away in the previous test, and we use them as well. And what we want are multiple tests so that we will not make mistakes. And so the red cells are, there's a lot of them in the blood. This just shows you the size of them. They're very little flat disc. They're about 2 microns. That's a small measure. And they're about 7.5 microns across. So what's known in the literature is something called red cell deformability. And that's because these red cells have to deform in order to go through the capillaries. Capillaries are smaller than the cell. And they have to stretch out and go through the capillary. And you can measure this deformability as how well do they deform. And it's found that in sickle cell disease, diabetes, and so forth, most interesting sepsis, that they don't deform as well. This could be, and we haven't figured it out yet, that it could be that there's an oxygen. It causes less oxygen to be delivered and less CO2 to be picked up. That would also contribute to fatigue. There are some problems in the blood flow as well that have been looked at from a total different point of view, but we haven't figured it out. So this is the device, one of them. We're trying lots of different devices, and what you can see is it's a tube that comes down. This is just very much like your blood flow. There's big veins and arteries, and then they come down to something very small, the capillaries, that the blood has to go through. So the tube that we're using is just a fabricated tube. It's not a capillary. But it's 5 microns, and that's less than a 7.5. So in order to get through, they have to deform. And we can simply measure how fast they go through, from a healthy as well as a patient. And there's a difference. So this is the possibility that we can take the blood and we take the right cells for one test, the red cells for a different test, and we look for consistency. And this will help, maybe this will help prevent us from getting a false positive, false negative. I just wanted to show you, this is another device that we can use. And what this is we're trying to make these very non-invasive and simple. So this is a test where you look at the nail bed, and you put a little oil on it that makes the nail transparent, and then you look at it through a microscope, and you can see these capillaries under the nail bed. And you can see the red cells flowing in that capillary. So we're going to try to set up an automated way to capture that and look at the rates. This is a little complicated, but it might be a very simple measure. And it could be a very quick thing, and we can build one of these very inexpensively, I think. So let me switch to treatment. I think we have some diagnostic tools. We'll continue to work on those. So the idea behind this is to screen the patients with the biomarkers before and after treating with drugs that maybe help the patients. Now we don't know. This is just looking at blood. So we could have a drug that is very effective at treatment and has no effect on the blood. But we'd like to know whether it does or not. We can also do this in vitro by taking the blood, treating it with the drug, and running the test. And if we found something that improved those, maybe they'll help the patient. So this makes the testing much faster and you don't need patients. So we found 2 compounds doing this in this way, doing it in the tube. One of them was copaxone, which was suggested about Robert Navio, and because there was a patient that was using it that was initially diagnosed with MS, if I understand it correctly. And this is an MS drug. And then she found out she didn't have MS, she had CFS. So why did this drug help her? And unfortunately, she became allergic to it and had to stop. It's a random copolymer of glutamic acid, lysine, alanine, and tyrosine. These are all amino acids. Simple. And it's simple to make. In fact, we got the patent, we looked at it, and we synthesized it, and it looks exactly the same. So it's easy to make. And so, since it's easy to make, it's also going to be easy for us to test. But part of it is important. And so, if we put this into our nanoneedle assay, what it shows is the fact that it greatly reduces the signal. It's doing something to the white cells. And there's 2 curves here, a blue one, which is a small dose, and then the green curve is the concentration of copaxone that is used in the patients. Problem with this drug is that it has to be given by injection. That's troublesome to some of the patients. It's possible that we could figure out what is important in this drug, and it was designed to be a drug that looked and mimicked the antigen site for the antibodies that are in MS. So it's supposed to look like a protein. We don't know that that's what it's doing, because what is it doing affecting the white cell population? There's no nerves here. So we might be able to make it smaller. And if we make it smaller, we might be able to get it absorbed under the tongue. That's just a guess. But Bob Navio also suggested cerumen, and we tried that. We've just done a few experiments with that. We tried it at the concentration that would be used on the patients. It does have an effect. It's not as dramatic. Now, this is just the white cells. It may not have much effect on the white cells. This has no reflection on what it will do to a patient. Neither of these do. It's just information. And then we looked at another compound called SS31. It's in clinical trials right now for congestive heart failure. It basically repairs mitochondrial membrane. Now that is more interesting. And I've talked to Bob Navio about this one as well, and there's a good shot that this might be something effective. Because the pumping of the sodium out requires energy, the energy comes from the mitochondria. So there could be some problem with the mitochondria, and if you repaired that damage, you might have an effect. And if you look at that, it really makes the white cells look like healthy, just by adding it. So this is a very encouraging one, but at the moment we can't get access to it to try it on a patient. It is almost, patent is almost expired, so it probably can be made available soon. And it's just 4 amino acids. It's not complicated to make. So we want to now expand. These are just the first trials of things that are candidates. We want to go through sort of random screening, and we have in our freezer all of the FDA-approved drugs. We've screened them all on yeast, and we know what they do in yeast, and there's a lot of similarities with that in human. We even have a lot of humanized yeast. So we can screen with using these kinds of assays. And I'd like to even screen natural products as well. So we're looking to get a set from China and from India. Now, in the course of doing all these things, we've, in our investigations, looking at what's going on with the patients, we decided to also look at the metals. Now, there's two basic types of metals. There are metals that are absolutely required for your health and life. For example, iron is necessary to be in hemoglobin. And if you don't have iron in the hemoglobin, you won't live. So there's a lot of metals like that that are very important carry-out reactions. And then there are the metals that are not so good to have, like cadmium and arsenic and mercury. So in the course of looking at this, and the way you do this is you simply take a blood or hair. This is an example of hair. The reason for the hair is that it's really easy to ship. You do a clip of hair, put it in an envelope, and mail it to us. So we can get hair from all over the world. And they don't need some phlebotomist to come in and take a blood sample. So the hair comes from cells. Now, it also can get contaminated. So we have to worry about the fact that it get contaminated from the environment. But you can also look at, some of the people we can look at blood when we get a blood sample. Interestingly, these are ME/CFS patients. And we've done more. This is just one small plot here to show you the connections. And about a third, 20% to 30% of the patients have mercury. And it's above the what's considered the WHO safety level. And our suspicion of why they have mercury is because they have digestive issues. And I'm mentioning this to you because autism has some digestive issues. And fish is easier to digest, at least the patients think so. Plus it has omega-3s, which you think is healthy. And so in fact, most of them have way too much omega-3s as far as I can tell. They overdo. And I think that our guess is most of these mercury is coming from the fish. So this is just a warning to be careful with your fish. If you eat a lot of fish, test your mercury. Now mercury is a toxin in itself, but it does a second thing. Mercury binds selenium. Selenium is one of those essential compounds. It's not quite a metal. It's in the transition zone between metal and non-metal. But it's involved in the enzymes as well. And you can see every time the mercury is high, the selenium is low. But there's one patient that has a lot of aluminum. There's another patient at the very bottom coming from Finland. This was a hair sample that has a lot of uranium. And we were shocked by the amount there. It seemed very high given that the reference range it's 100 times the reference range. However, after collecting even more samples, we have found patients that are 10,000 times that in uranium. Now, the patients that are extremely high probably come from well water. And the well probably is leaching out uranium from the environment. The problem is the uranium in the environment is radioactive. And it's an alpha emitter. Internally, an alpha emitter is very carcinogenic. And in fact, the patient who sent this was later diagnosed with cancer. So probably not a bad idea if you use well water to get it tested. You could have very high selenium, you could have arsenic, you could have uranium, you could have anything in it, and it's not that expensive to test. And one test will do everything. The problem with selenium being low is it's essential for a number of very important reactions. 3 of them are antioxidants. That means they scavenge out oxidative damage. and there appears to be a lot of oxidative damage in the patients. It's also involved in conversion of T4, that's a thyroid hormone, to T3. Many of the patients are low in T3. So we're going to do a much bigger study on this if we can get some funding for it. But so now we've also done a large number of tests. I'm not going to go into detail, just showing you there's a lot of tests we've done, a lot of data. It's because we can run tests that nobody else can run. I think we probably have more data on these patients than any other patients in history. And we're still looking through it. It's a lot of data. And I was going to give you some highlights that I thought might be useful. One is that the patients are, these are severe patients that we've done, excuse me. So we've only done the bed-bound patients. Nobody's ever studied them before. We go to them, take the samples. They don't come to us. So all the patients are low in something called indole. And so, of course, the first thing you do is say, wow, they're all low, maybe they have a mutation. Well, no, because that metabolite is not made by human. It's made by a bacteria in the gut. And then we look for the bacteria in the gut, and it's not there. So this is the case where, yes, the microbiome is probably having an influence, but it's not because it has something bad in it. It has something that's missing. Now that would be a lot easier to treat in a way. You don't have to get rid of the existing flora. You just simply have to add this one. Now my guess is it'll go away again because it's missing in all of them. So we'd have to try to figure this out, and also one could even give indole if it gets through clinical trials. The other thing we found in the blood is, in all the patients almost, is high lysine and high hydroxyproline. I'm just pulling this out from thousands of things we've measured. Those two things are involved in collagen. So it appears that there's collagen degradation going on in the patients. Now, we found this just simultaneously with the realization that a number of physicians had found that in a number of patients, not all, that their cranium has fallen down because of lack of support by lack of collagen, and their cranium has fallen down onto their spinal column, and it impacts the brainstem. And now some of these patients have now raised their head permanently by putting metal rods screwed into their skull and screwed into their spinal column. And all of their symptoms go away. They're instantly cured. Now, the physicians say, I don't think that's true for everybody, but why do they have consistent symptoms with the people you think don't have them? Don't know. You have to figure it out. And I'm going to move forward because I'm running out of time. And I want to go to something that I think would be very useful. We've looked for infectious. We haven't found anything. We keep looking. But I want to go through some of the sequencing results. And that is that we have found that all 20 of our patients have a mutation in this gene called IDO2. We've done it 46 additional patients. They also have the mutation. Now, the genesis cell is, that's irrelevant. It's a common mutation. Well, there's 4 different ones we look for. And they make a defective gene. And they're common in the population. They cannot possibly be relevant. But statistically, it's kind of like flipping a coin and doing it 70 times and it comes up heads every time. You worry about the coin. So we begin to explore this and think about what it might mean. So here's the pathway. And it's interesting because there's 2 genes, IDO1 and IDO2. They convert tryptophan ultimately to kynurenine, and kynurenine to something called NAD, and NAD is required for energy production. Kynurenine controls the immune system. It also controls inflammation. Also, tryptophan leads to serotonin. So this makes you worry in these pathways. These are all relevant to what's going on in the patients. So we begin to look at the literature for this. And also the biochemists tell us that, oh, this can't possibly be involved because IDO2 does the same thing that IDO1 does, and so you couldn't possibly have any effect of the mutation. Now, these are people that are very narrow in their focus, either just genetics or just biochemistry. I'm fortunate to I work in both, so you want to look at that. And here's one problem. And as far as we can tell, this is not something that people have recognized before. So the biochemists look at the pathway for making tryptophan to kynurenine. And the surprise is, and you can see on this graph in the blue, is what IDO1 does, is that it increases the flux of making more and more kynurenine as you increase tryptophan. That's exactly how all enzymes should work. But when you get a critical concentration of the substrate tryptophan, it becomes an inhibitor. So it's called substrate inhibitor. In other words, if you have high tryptophan, you can't make the products that it's supposed to make. And the only way to get rid of the tryptophan and lower it is to consume it, which you can't do because you're blocked. Now, in the body, you have another gene called IDO2. Not very essential. However, if you get trapped, it's the only way out. And the only way to reduce the tryptophan is for IDO2. Now, that isn't going to work if you have a mutation. So this is a case of a gene that may never work your entire life, or it may work occasionally to keep you out of this trap. So we call it a metabolic trap. And I think it points out an interesting observation. The biochemist alone would disregard this because you have IDO2. They don't think about you have a mutation. And the geneticists look at this and say, you have two genes, IDO2 is not necessary. Mutations, a lot of people have mutations, they're obviously fine, and never explore. So you've got to have a very open mind when you think about this stuff. And Robert Fair has been the one who sort of put this together. He's what we call a system theorist. He's has a degree in electrical engineering from MIT, and then decided to take those same technologies of like electrical flow and apply it to biochemistry flow. And he has all sorts of models and programs to do that. And so he can model everything, will this work? And so these are just some of the key points about that. And it's an example of really two states. There's a healthy state and a disease state. And you can simply be flipped from one just by this one enzyme. We don't know if it's right yet or not. I'm just pointing out what you have to think about. And if you get into this other state, you can't, in these circumstances, if you have effective IDO2, you cannot get out. You can model that. And accidentally you could get out, but it might take 100 years. That's not a good treatment. So, looking further, there are about 80 enzymes that are substrate inhibited. Many of them have this two enzyme system in them. So there could be a number of metabolic traps affecting human health, and what they would be are chronic diseases. Because once you have them, you can treat the symptoms, but you can't cure it. If you figure out what they are, we can probably develop a drug that will push you back and get you back into the stable state of health. Anyway, that's what we're working on. And I think the best way to do this kind of work is not to be in my lab, keeping secrets, and not telling anybody what we do. I tell people what we've discovered, even if it might be wrong, because we'll keep testing things. And we need a research community to do this, not some individual labs that are competing. And a colleague of mine from Harvard came in and he was saying, you give all this stuff away, and you make it possible for other people to do it. You're not competing with anybody. How do you get motivated if you don't compete? Disease motivates you, not the competition. Thank you.